My name is Dr. Mikey Ross, and I'm a postdoctoral researcher at the Scottish Association for Marine Science. I'm also the manager of the culture collection of algae and protozoa. In terms of my background, I started my career as an undergrad at the University of Stirling in marine biology. I then continued into doing a master's in sustainable aquaculture there. My project was trying to improve broodstock and the kind of natural restoration of the native oyster. So I was looking at different algal diets and preparations for them, so to try and improve their feeds to make them breed more effectively and have stronger offspring. So that was my first introduction to algae. From there, I then started as a technical post at SAMS, the Scottish Association of Marine Science, on a project called Biomara. This was looking at trying to get renewable bioenergy from algae, so both microalgae and seaweeds. So looking at their kind of lipids and carbohydrates and things like that there. From there, I then worked as a member of technical staff down at the University of Swansea, who were kind of working at scale. We've got facilities there, which are a few hundred to a few thousand litres. From there, I started a PhD in Edinburgh in collaboration with SAMS as well. And this was looking at wastewater treatment with a type of algae called Cladophora, which is often considered a pest. It forms these big population booms in the Great Lakes, for instance. So I was looking to remove nutrients and heavy metals from water. And from there, I then started a postdoc here at SAMS, and I've been here ever since. So working on a whole variety of different algal-related projects, from bioenergy, high-value products that can be used in cosmetics, nutraceuticals, pharmaceuticals, right through to feeds, foods, bioenergy, you name it, probably investigated somehow. And then in September last year, I took on this role as CCP manager. CCAP, so that's the culture collection of algae and protozoa. So we are hosted in the Scottish Association of Marine Science, SAMS, and the West Coast of Scotland. And SAMS is part of the larger kind of UHI, University of Highlands and Islands, which has got 11 campuses dotted around Scotland, and UHI is part of SULSA now. CCAP, we are funded by NERC, the Natural Environment Research Council, under the kind of UK Research and Innovation umbrella. So we are publicly funded, and we are what's known as a national capability. So our whole remit is to provide services and kind of foster collaboration within the algal community, both commercially and academic in the UK and internationally. As a national capability, we are what's known as a biological resource centre. That's quite a strange term for some people, and we often describe it as being like a botanical gardens, but whilst we don't deal with plants or seeds or trees, we deal with algae, cyanobacteria, seaweeds, pathogens, protozoa, so a whole huge diversity. CCAP, as a culture collection, we're one of the oldest in the world. Our oldest strain dates back from 1890, and we've kept it in continuous culture since then. And it's had a huge deal of research, and that is called Chlorella vulgaris. We've got over 3,000 different strains, representing over 1,000 different species. So we've got a huge biodiversity across the whole taxonomic tree of life. Yeah, we provide a huge variety of different services with the collection as well. So it's probably best known internationally for the provision of these strains, which we send out all over the world every week. But yeah, we provide a whole range of different services, so molecular services, so people could send us cultures and we can barcode them, find out their identity. We are involved in education and consultancy, outreach activities. Maintaining 3,000 different strains is a huge amount of staff effort for that. So we've got about half of them in long-term storage under cryopreservation. So we've got a legacy of that. And collaborative research as well, we're involved with too. In addition to CCP services, which I just touched upon, what we were finding with time was that we were getting a lot more requests from our customer base for larger volumes of algae. So with the culture collections that we've got, we would typically send out small volumes, like 10 millilitres up to 50 millilitres cultures, so that people can begin working there and scaling up. But we were getting increasing number of requests to have larger volumes of culture, which would be more representative of pilot scale systems or commercial activity. We were getting more and more requests for more advanced molecular work, so barcoding and taxonomy, and then moving into whole genome sequencing, which is becoming increasingly kind of prevalent in science. and a lot more analytical things as well. So find out what the algae are producing, whether it be pigments or fatty acids or proteins. And so late 2020, we received capital funding from NERC, our funding agency, who provided us with money to set up this new centre called CCP ARIES, the Algal Research Innovation Environmental Science Centre. So this is kind of triphasic. It's got three different aspects to it, which kind of satisfy all those different needs. So we've got 4 new what are called photobioreactors, basically kind of vessels in which you cultivate algae. So you provide light, air, CO2, and you can put nutrients. And it's a closed system. So what you're growing in there is what you're growing, not something else, bacteria or contamination. So we've got 4 of those, which are 70 litres each. So 280 litres in total. So that's the cultivation side, but you've got quite a large volume of liquid in there. We've got downstream processing, so ways in which we can harvest those systems as well, using membrane filtration and centrifugation. We've also got new analytical equipment. So we've got what's called a GCMS, gas chromatography mass spectrometry, which allows you to do metabolite analysis to find out what fatty acids are in there, what vitamins, anything really. So quite sensitive equipment. We've also got other equipment to help characterize what the algae are producing too, as well as new molecular equipment to kind of automate systems or build different libraries so you can kind of better curate the different algae or whatever we've got. So the ARIES system, it's these 3 different aspects. You can use it in its entirety, or you can use individual components. So if you just want a different pigment analyzed from a certain type of algae, we can do that. However, if you want to write a whole research grant, we can do that too. And yeah, it's there for the community. We're not really charging kind of commercial rates. We're there to kind of really foster collaborations and things like this. CCP, we are publicly funded and we are all about collaboration. That's kind of our bag. So yeah, we're part of a variety of different networks. So we're part of the British Ecological Society, Algae UK. We're part of the UK Biological Resource Centre network. And recently I was in Germany for the European Culture Collection Organisation meeting, and I met some really cool people there. So they're all kind of like-minded people. Whilst CCP focusing on algae, there were other culture collections dealing with bacteria or fungus or viral cell lines and things like this. So I met some people from an institution called DSMZ in Germany, and they're probably the largest bacterial culture collection in the world. And they've recently set up this really cool platform called Media Dive. And so it's essentially a kind of repository for all the different types of growth media or growth formulations that you need to cultivate these different microorganisms. And chatting to those people there, we integrated CCP's kind of media recipes there. So we've got over 100 types that we grow all our different culture collection in there. So it's a kind of free to access point there. Perhaps in terms of CCP's collaborative research, our flagship project that we're probably involved with now is the Darwin Tree of Life. You may have heard of it. It's part of the larger Earth Biogenome Project. And the Darwin Tree of Life, that's aiming to get the whole genome sequence of 70,000 eukaryotic organisms native to the UK and Ireland. CCP, we're involved with the Protistan group, and we're providing a few hundred different types of microalgae and seaweeds. So it's going to be huge. It's picked up quite a bit of traction in the news, and I think it's going to be a really kind of sea changer, pivotal project for the next foreseeable future. It's going to be groundbreaking. In terms of CCP ARIES, we've just finished, or about to finish, a project with a company in the States called Living Ink, and they're a company trying to produce sustainable inks. So we had a collaboration with them a couple of years ago, funded by Algae UK, and our business cards are now actually printed on algae-based ink. This time we were trying to get a blue pigmentation from the cyanobacterial cultures for them. So pretty neat. CCP ARIES was launched in April this year. So kind of future plans for that is just to try and build momentum and kind of develop that, make it a bit more in people's conscious, and develop that and have a good track record with it. We'll have a project in January. So one of my PhD students, Ellie Wood, will be cultivating her algae called chlorochromis, which produces different carotenoids, so highly pigmented molecules. They're the things that give carrots the orange color. So she'll be cultivating in that. For CCP more generally, so we're funded by NERC over a 5-year installment. So we're kind of coming to the end of our current commission. So we'll be looking ahead into strategizing for the next 5-year period, 5 to 10 years, and applying for funding so we can keep providing these services to the community and collaborative efforts. In terms of collaboration, so CCP, I will be presenting at the Scottish Natural Products Summer School, which Alison here is kind of helping to organise. So with the ARIES Centre, with the different equipment we've got great capacity to analyse all these different metabolites. So it's really kind of key to our facility for all the different researchers in that body of work. So really looking forward to that and engaging with a kind of new audience there.